All right, let's talk about something I think we've all been through. Staring at a brand new, empty qPCR plate in the CFX Maestro software and thinking, okay, where do I even begin? Well, you're in exactly the right place. In this explainer, we're going to walk through step by step how to turn that blank grid into a perfectly designed experiment.
Trust me, by the end of this, that blank plate isn't going to look intimidating at all. It's going to look like a canvas, ready for your next big discovery. So let's jump right in.
So here's our game plan. We've got 5 main sections. We'll start with the blank canvas and those first critical steps. Then we'll get into populating the plate with our samples. After that, we'll build our standard curve, make sure everything is high quality with controls, and then cross the finish line with a few pro tips. Okay, section 1. Before we can even think about adding samples or standards, we've got to get our digital workspace set up correctly. This is the foundation for everything that follows. This initial setup is really just 3 simple clicks. First, you create a new plate file. Next, you tell the software what you're working with, usually a 96 or 384 well plate. And finally, you have to choose the right scan mode for your particular experiment.
So this is a big question, right? Which scan mode do you pick? Well, it's pretty simple. If you're doing a standard experiment with a single dye, like Cyber Green, that Cyber/FAM option is your go-to. But if you're running a more complex multiplex assay with multiple different probes, you have to select all channels. This tells the instrument to collect data from every fluorophore. Getting this right from the start is absolutely crucial.
All right, our digital plate is ready to go. Now for the fun part, adding the core of our experiment, which is, of course, our unknown samples. And the software makes this super methodical. You just highlight the wells you're using, tell the software that they are unknowns, assign your fluorophore and the gene you're looking for, we're using IL-1B as an example here, and then you give each sample a unique name. This is how the software keeps track of everything for the analysis later. Now, you're almost never going to run just one well per sample, right? You're going to run multiples. And these are called technical replicates. They're not just for looks, they are a vital quality check. They tell you how consistent your results are and, honestly, how good your pipetting is. And here is the golden rule. You absolutely have to remember this. For the software to understand that a set of wells are technical replicates of each other, every single descriptor has to be identical. The fluorophore, the gene name, the sample type, and the sample name. They all have to match perfectly. If even one thing is different, the software won't group them correctly. Okay, so our unknowns are on the plate. But how do we know how much target is in them? That's where the standard curve comes in. Think of it like a ruler that we're going to use to measure the quantity in all of our unknown samples. And setting this up is actually one of the fastest parts of the process. You highlight all your standard wells, change the sample type to standard, define your replicates, and then you use this magical tool called the dilution series. You just plug in your starting concentration and your dilution factor, and boom, Maestro does all the calculations for you. It's a huge time saver. Now, a quick heads up here. When you use that awesome auto dilution feature, there's a little rule baked into the software you have to know. It requires a minimum number of technical replicates for each point on your curve, and that number is 2. So why does it do that? Is it just being difficult? No, not at all. It's actually a good thing. The software is basically nudging you, the scientist, towards better experimental design. By enforcing this rule, it makes sure your standard curve is robust and that your final quantification is going to be as accurate as possible. It's a best practice that's built right in. We are in the home stretch now. We've got our unknowns, we've got our standard curve. Let's add those final touches that not only ensure the quality of our data, but also unlock some really deep analytical insights. First up, the No Template Control, or NTC. This is non-negotiable. This is your contamination check. If you get a signal here, you know you have a problem somewhere, and setting it up is just as easy as anything else. Select a well, pick No Template Control from the dropdown, assign your gene, and you're good to go. Now, beyond the basic controls, CFX Maestro has this incredibly powerful feature called biological groups. This is what lets you go from looking at just individual samples to analyzing entire experimental conditions, like comparing all of your treated samples against all of your untreated samples in one go. So for example, you could grab all the technical replicates from sample 1 and sample 2 and tell the software, hey, all of these belong to my untreated group. Then you could do the exact same thing for samples 3 and 4 and call them the treated group. This makes your data analysis later on so much more powerful and easy to interpret.
And just like that, we're at the finish line. Your plate is designed, it looks beautiful, let's talk about saving it. And then I've got one last pro tip for you that you're gonna love. So, once everything is perfect, you just go to File and Save. But here's the real magic. That saved file is now your template. The next time you run a similar experiment, you don't have to start from scratch. You just open this template up, make a few small changes, and you are ready to go in a fraction of the time. And finally, here it is, the one tip that will literally save you so much time at the bench. Get this. You do not have to finish setting up your plate in the software before you start the run. You can physically load your plate into the machine, hit start, and then go back to your desk and set up this virtual layout while the instrument is running. It completely separates the software work from the machine's runtime. It's a game changer. And there you have it. You've gone from that intimidating blank grid to a perfectly designed, rock-solid experiment, all ready for analysis. The technical part is done. Now for the really exciting question, what story is your data about to tell?